This PowerPoint presentation is about the big event. What is this big event? It is the event of fertilization. What is fertilization? It is the union of the sperm with the ovum. Do we really need the help of questions? The answer is, of course. Without them, we cannot extract a lot out of this image, which is worth 1,000 words. First, sperms are ejaculated into the vagina. Let's talk about the sperms. The sperms are living in a very suitable environment in the seminal fluid, and the number ejaculated into the vagina is a huge number. It is 200 millions to 300 millions. The question is, do we need such a number? The answer is yes. Why? Because lots of sperms are going to be sacrificed along the way. Is the way long? The answer is yes, it is very long for the sperm. And the first difficulty is that the environment of the vagina is quite different from that of the seminal fluid, and therefore the sperms will face difficulty number one, and that is the living in a different environment. Then sperms will swim through the very energetic movement of the flagellum, and they go up the cervix, the part that connects the uterus to the vagina, and they get into the uterus. Is the environment of the uterus good enough for the sperms? The answer is no. That is difficulty for the sperms to adapt to, and millions of sperms will die in the uterus. Then the viable sperms will move towards entering the fallopian tubes. Is the environment of the fallopian tubes good for the sperms? The answer is no. Why is that? First of all, chemically it's different. Secondly, there is a very important factor, which is what? It is the movement of the cilia of the lining of the fallopian tubes. What is the direction of movement of these cilia? It is from the lateral side towards the uterus. And the sperm wants to go to the lateral end of the fallopian tube. Therefore, they swim upstream against the movement of the cilia, and that is another difficulty. Then these sperms, they need to be modified so that they can enter the fallopian tube and do the job of fertilization. They are not good enough for this process. Therefore, they stop at the beginning of the fallopian tube and start to have changes. These changes happening at the beginning of the fallopian tubes is called the process of capacitation. Without it, sperms will not survive and will not be able to fertilize the egg. This is something we will be discussing later on. And when they are capacitated, sperms will travel upstream towards the ampulla near the febrile end, and there they are going to meet the ovum. What is this image telling me? It is showing me the three-dimensional image of sperms, showing the head, the middle piece, and the tail. Where are these sperms? If you see the floor of the image, it is smooth and a little bumpy. That is the vagina. So these are sperms in the vagina. What is this image showing? I can see sperms, seeing the head, the middle piece, and the tails, and I can see that the environment is different from the previous slides, and this is a scanning electron microscope of sperms in the uterus. The background or the environment the sperms are in is the internal wall of the uterus. Question: what is this image telling me? It's an image of sperms looking from the left. You can see a diagram of the sperm when they are in the seminal fluid, and look at the head of the sperm where you can see attachment to this head. If you go to the right, you will see an ejaculated sperm in which more chemical materials are attached to the head of the sperm. Now, when the sperm gets into the female tract, you will find that the attachments, the green ones, are being lost, and the head is a little bit smaller. This is a capacitated sperm at the beginning of the fallopian tubes. This is an exercise image for you, and you must be able to ask questions and extract the facts from this image. Without questions, you will just see this image and you stand still, and without the motivation of the questions, you cannot learn anything. Now, at this stage, we have to stop and see what we have collected about the process of capacitation. Capacitation is a process. Now, we want to collect some basic facts that are very essential for us. What is capacitation? Is an activation, a process that will take place mainly on the head of the sperm, preparing the sperm, first of all, to pass into the fallopian tube, and most importantly, it should be able to fertilize the ovum. capacitation begins once the sperm enters the upper female genital tract. What is the upper female genital tract? It is the upper part of the uterus just before entering the fallopian tubes. And what does capacitation, most important process? The most important thing that should happen is that at the head of the sperm there is a cap called a chromosome. It is equipped with all the necessary enzymes to pass through the wall and the layers of the wall of the ovum. Now, capacitation prepares the chromosome to do its function. The main function is to penetrate the outer layer of the ovum for the sake of fertilization. And look at this fact. No capacitation process, no fertilization. It is very logical. Capacitation prepares the sperm, the head of the sperm in particular, to pass through the wall of the ovum, and no capacitation, no fertilization. We need more specific details about capacitation. Where does it happen? It happens at the head of the sperm, and in addition to that, preparing the chromosome, the chromosome is going to be more permeable. There are two major changes in the sperm. Something which we already had an idea is the removal of proteins and other materials covering the head of the sperm. And secondly, it's the tail, the flagellum. The flagellum is the main engine that the sperm used to advance in the female genital tract, and through the movement of whipping, just like a fish. And this flagellum has to be more energetic, and it beats faster, and even the head is going to move right and left. What is this three-dimensional image showing us? It is showing that the sperms are getting into the fallopian tubes and traveling towards its lateral end. Questions are very important. Not only the questions that I ask myself, but you have to ask yourself even more questions. What is this diagram showing? It is showing that the few number of sperms that have reached the ovum are approaching the ovum, the outer shell of the ovum. And we know now the layers that cover the real oocyte. What is this image telling me? It's a diagram showing that one sperm has reached the outer cellular part, the corona radiata covering the ovum, and it attempts to pass through it. What process enables the sperm to pass? It is the very energetic movement of the flagellum, that's the tail, and the right and left movements of the head. This is where it is happening. Here is our friend helping us to ask questions. What is this diagram telling me? It is telling me that this sperm has already passed the corona radiata and it is about to touch the zona pellucida. What factors enable this sperm to reach this stage? It is the movement, the very energetic movement of the flagellum and the movement of the head of the sperms. What is the event inside this yellow circle? It is step number 2 after penetrating the zona pellucida. The head of the sperm is touching the zona pellucida. What structure becomes very important and starts acting now? It is the acrosome. Now, if we ask ourselves, what is the acrosome? We know at this point of time that it's a cap on the head of the sperm full of enzymes that will digest the zona pellucida. And this action is going to happen now. When the head of the sperm touches the zona pellucida, zona pellucida is not an ordinary structure. Now, we know that it was secreted partly by the ovum and partly by the corona radiata, and not anything is going to pass. There is a special door for the sperm. That special door is a receptor. This receptor is called ZP3. Now, the head of the sperm should come and fits the receptor. If it doesn't, forget about the whole process. It must fit this receptor. What is this? This is a diagram showing what? It is showing that zona pellucida covering the ovum has receptors, and these receptors are of one type, and it is receptor ZP3. And I can see that the head of the sperm has already approached the zona pellucida, and the head of the sperm is fit into the receptor. Therefore, the key was introduced into the door. The door is going to open. what is happening in this yellow circle? You can see that the head of the sperm along with the receptor, they are given a red shadow. That means here is the process of action. Now you know where we are and what is happening. Now, if you look into this oval yellow shape, you will find that the red shadow is not only around the acrosome as it was viewed in the previous slide, because the acrosome is now releasing its enzymes, and these enzymes are starting to affect the zona pellucida. Therefore, the zona pellucida is here included in the red shadow. Question: what is happening here? The head of the sperm, with the help of the acrosome, with the help of fitting on ZB3, the head of the sperm now entered into a space between zona pellucida and the ovum. This is called the perivitelline space. And the head of the sperm is touching the plasma membrane of the ovum. The question now is, as the head of the sperm touches the plasma membrane of the ovum, and here is the head of the sperm has touched and indented the plasma membrane. The plasma membrane of the ovum is going to change to what is called secondary membrane. What was the primary membrane? The primary membrane was before the touching of the head of the sperm. Now, what is the difference between primary and secondary is something we are not going to address. But now the plasma membrane is different. The question now is, what is happening after the sperm has penetrated corona radiata, the zona pellucida, and touched the plasma membrane of the ovum? What about other sperms? These other sperms, there are few. When we say few, we say in the early hundreds, out of the 200 to 300 millions. Now, here is a sperm that has reached the ovum, but that's it. No way you can get into the follicle or the ovum anymore, because the other sperm has already entered. Therefore, this sperm is going to be blocked, and blocked very quickly. And this blockage prevents a process called polyspermy. And polyspermy means that more than one sperm enters to face the ovum, and this is a forbidden process. We need to ask questions about what is happening here in an attempt to secure that nothing passes into the ovum other than the winner sperm. The ovum is going to change its permeability, stimulated by the touching of the acrosome of the winning sperm, and with this change of permeability, calcium ions will flow into the cytoplasm of the ovum. What is going to happen and what are the events? Event number one is that when the head of the sperm touches the plasma membrane of the ovum, then the only structure pushed into the cytoplasm of the ovum is the nucleus. The nucleus here is haploid, containing half number of chromosomes, plus some cytoplasmic contents like mitochondria. And the plasma membrane of the head of the sperm, the middle piece and the tail, they are not allowed to get in. The second event happens in the ovum. Meiosis II is going to be completed, and it has two. So the end result is a haploid number of chromosomes ready to be completed by those of the sperm. And you had prior knowledge of meiosis I and meiosis II, and in case of the female ovum, there are polar bodies, and you can see in the upper right the polar bodies that resulted from the process of meiosis. With the help of the questions, we can be able to extract data from this diagram. First of all, the nucleus of the sperm has changed its shape to male pronucleus, and it is the same as that of the ovum. So we have two pronuclei inside the cytoplasm of the ovum. These two pronuclei are approaching each other, and the big event of fertilization is about to begin. This is the nucleus of the sperm. The nucleus is going to start losing the nuclear membrane, and the chromosomes will unwind into thread-like structures and then rewind to form the chromosomes again. The oocyte, which has not completed meiosis II, is going to complete it. And you can see the microtubules trying to arrange chromosomes, and these chromosomes of the oocyte is going to be reaching the midline, and then the sperm nucleus and the oocyte in nucleus, they are going to approach each other and fuse, and the chromosomes are going to be aligned and combined. Now, the two nuclei, the number of chromosomes now has doubled, half from the sperm and half from the ovum, and this union is going to form the first cell that will form the human body. This is the big event, and this is the most important. This was the target of the whole of gametogenesis and all the journey of the sperms, and it comes to its final end, and that is formation of the zygote. Once the zygote nucleus is formed, there will be a zygote cell containing 2n numbers of chromosomes, and very soon it is going to divide into two cells. It has no time, it has only 9 months to create a human being. The two cells are going to divide quickly into 4 and then 8 and then fertilization takes place at the ampulla of the fallopian tubes, which is the dilated part near the ovary end, and it is going to be transported along the fallopian tube in a way that you saw in the film, and then it reaches the junction between the uterus and the fallopian tube, and this is around day 5. Now, in day 1, it is 2-cell stage, day 2, 4, then day 3, 8, day 4, 16, and this is a stage called morula, where the cells are packed together, surrounded by zona pellucida. Now, in day 5, it is going to reach the uterus, and the morula is going to have a space between the cells called the blastocele, and the whole structure is called the blastocyst. It is going to form a collection of cells on one side called the inner cell mass. This inner cell mass is going to form the human being, and the outer cells are called trophoblasts. They form the structure that will feed and nourish this structure. This is one of cross-section diagram of the blastocyst. Here is the inner cell mass. Cells are also called embryoblasts, and here are the trophoblasts that surround the blastula, and the cavity is called blastocyst. What is this ball? This is a three-dimensional view of the blastula, and the cells we are seeing in front of our eyes are the trophoblasts. What is inside this spherical structure is first of all an inner cell mass that will form the embryo and the human being, and a cavity that is called the blastocyst, which contains fluid for the sake of nourishment. The bottom line is this: a modular collection of cells called embryoblasts. The morula is going to form a blastocyst. Blastocyst is an inner cell mass and a cavity. The inner cell mass is going to form the embryo, which develops into the human being, and the trophoblast is going to form structures that will nourish and feed the inner cell mass. Here we can see the blastula, where it has the inner cell mass on one pole of the ball. Then we have the cavity, the blastocoel. Then we have the trophoblastic cells all around, and the zona pellucida is still around, but it is going to degenerate very soon. This is a diagram of the blastocyst, made of inner cell mass, the blue cells, and a circle of trophoblasts, and the cavity is blastocoel. Now, this is what is going to happen to the blastocyst. These cells of the inner cell mass are going to divide and produce a single layer of columnar cells lining this surface of the inner cell mass and lining the blastocoel all around with cuboidal cells. Now the cavity was called blastocoel, but now when it is lined with these cells, it is called Yolk Sac. This is the invading system that helps the embryo to invade the endometrium. This is the embryo, and this is the cytotrophoblast layer, This cytotrophoblastic layer is going to divide and send to the outside cells that will form syncytiotrophoblasts. These cells are going to lose the cell membrane and become a large mass of cytoplasm with many nuclei that do not divide, and this structure of cytotrophoblast and syncytiotrophoblasts is called the chorionic membrane. Now, these cells, the syncytiotrophoblasts, is a large mass of cytoplasm. This large mass of cytoplasm will develop vacuoles, large ones. These are called lacunae. A single one is lacuna. Now, the chorionic membrane facing the embryo is called the chorionic frondosome. In this area, the syncytiotrophoblasts will develop much more than areas other than facing the embryo. This is a diagram showing the yolk sac, and the yolk sac is also called exocoelomic cavity. This is what is called ultrasound of the uterus, also called sonar. Now, this black circle is what is called the yolk sac. This is the first indication detected by the sonar or the ultrasound that pregnancy is progressing. That means there was fertilization passing through the tube, getting into the uterus, and this is going to be the first evidence of pregnancy has happened. Again, this is the invading system of the embryo, which is the beginning of the formation of the placenta. Now, this is the future embryo, and this is the cytotrophoblast layer, which is going to divide and gives to the outside the syncytiotrophoblastic layer. This syncytiotrophoblastic layer will develop vacuoles that are called lacunae. The invasive process is going to continue by secreting enzymes that are going to destroy the endometrium and pushing the embryo into the endometrium. This is an invasive process. If it continues, it is going to invade even the myometrium, which is not favored. Therefore, the uterus is going to produce a reaction to the invasive process. This reaction is going to be called decidua. This decidua stops the invasive process. The decidua is made of a modification of the endometrial cells by producing a dense layer. Let us look at this diagram and see what it tells us. First of all, this is the yolk sac, and this is the inner cell mass producing the bilaminar germ disc. The cells facing the yolk sac are the hypoblasts. The other cells of the inner cell mass will develop a cavity called amniotic cavity. The cells facing the hypoblasts are called the epiblasts. These cells are going to form the human being, and the cells facing the chorionic membrane is called this is a diagram of an implanted embryo into the endometrium. This is the epithelial lining of the uterine cavity, and here is the point of entry closed by a blood clot, and this is the embryo with its invasive system, the early beginning of the placenta. And this point of entry, when it is closed by a blood clot, few drops of blood comes down, and the lady thinks that this is a kind of change of the bleeding phase of the menstrual cycle. Let us write the very basic headlines of what happens in fertilization. First of all, there's the process of ejaculation of the sperms into the vagina, and it starts with 300 millions of sperms, and what reaches the ovum is around 100. In order that sperms can find its way to the ovum at one stage, it must be capacitated, it must be enabled to enter the ovum. And fertilization process, the big event, is going to occur in the fallopian tubes near the fimbrial end of the tubes. The main event that takes place to help the sperm to enter and penetrates the layers of the ovum is the acrosome. The acrosome contains an enzyme called hyaluronidase, and this is the basic enzymes that performs its action. And a sperm fuses with the ovum, a cell membrane, and it is only the nucleus and a few cytoplasmic organelles, especially mitochondria, will get into the cytoplasm of the ovum. Here is some information about the viability of a germ cell. Sperms, they remain alive inside the female genital system for about 3 days, around 3 days. The oocytes are usually fertilized within 24 hours after ovulation. They live shorter life than the sperms. When the oocyte is taken out of the female genital system, that is in vitro. In vitro means outside the human body. In vivo, it means when it is in its ordinary place inside the human body. This unfertilized oocyte, when it is taken out of the female genital system, it is usually preserved in a special environment, and these cells in vitro, they die within 12 to 14 hours, maximum 14 hours. So they have shorter life because they are outside their natural environment. Fertilization process requires about 24 hours. Let us summarize now the process of implantation. First of all, the zona pellucida that surrounded the blastula is going to degenerate. Then, when it degenerates, the trophoblastic layer will be the external layer of the embryo. It is made of 2 layers, the syncytiotrophoblasts and the cytotrophoblast on the inside. The syncytiotrophoblast on the outside has these basic facts. They are not cellular. That means that the cells are fused together, creating a large mass of cytoplasm, which has many nuclei. This is going to penetrate the uterine wall.